Now I want to take just a minute and talk about mutations. So I mentioned to you that the genetic code has those redundancies where more than one codon will code for the same amino acid. And that's a good thing when a mutation occurs in some cases because the amino acid that's coded for isn't changed, therefore the protein won't be affected. Now there's different kinds of mutations. So the first one is a point mutation, and a substitution would be an example of that. So for example, in this first example here we see A, G, C, G, T. Right here is an A, but if a mutation occurred and it's no longer an A, but is in fact a T, it's just a substitution. But notice we didn't have any change in the amino acid that it was coded for, so we call that a silent mutation, meaning there shouldn't be an effect on the protein product. Now, if we look at this next example, okay, what we see here is there's a C originally, but here we have a substitution of an A. And in fact, that does cause a difference in the amino acid that's coded for. So this depends. It depends on chemically how similar, how different are those amino acids. Sometimes it may not cause an effect, but sometimes it can have a detrimental effect on the functionality of the protein. Now, when we start to have likelihood of greater issues is when we have what we call a frame shift mutation. So if you think about if you're reading a sentence and you inserted a letter and that letter actually threw off all the rest of the sentence. So if you kept the number of letters per word the same, you inserted a letter and it would throw off the rest of the sentence wouldn't make any sense because none of that would be words. That's sort of like a frame shift mutation. Because these are read in groups of 3, if there's only an insertion of 1, okay, then it's going to throw the reading frame off. Now, down here we see some examples of frame shift mutation. So if we look at this point right here, we have A, G, C, G, but this TA that was originally there is actually going to be deleted or removed. So instead, we don't have that there. Instead, it just shifts this C, right, all the way over. So what happens is this first amino acid is not changed. But since it shifts the reading frame, every amino acid after that is likely to be different. So that is likely to cause a lot bigger issue in the functionality of that protein. And we would see the same thing, right, if we had an insertion that also threw off the reading frame. Now let's talk for a second about gene regulation. And by regulation, what I mean is which genes are being turned on and which ones are not, and how much protein is being produced by those genes. Well, we need to recall and understand that it costs the cell energy in order to produce proteins. So plus proteins are active and doing things, so we don't want to spend energy unless we need to, and there are certain times when we need particular proteins present and we don't, and we need to regulate their presence. So in prokaryotic cells, we're going to look at an example of something called the Lac Operon. So an operon is when several different genes are all under the same control of the same promoter. We call that an operon. And we're going to look at how this Lac Operon works because it's a very good example of how to look at when something is turned on when it's needed and it's turned off when it's not. So we'll look at that in detail in just a second. In eukaryotic cells, what I want you to think about is very complex because we have a nucleus and there's multiple points that this can be regulated. So for example, something called epigenetics. This is when the DNA, it can either be very tightly wound or it can be less tightly wound or more loosely wound. The RNA polymerase, if DNA is too tightly wound, just cannot get to the promoter, cannot begin transcription on a gene. So epigenetics is actually regulating how tightly wound that DNA is and whether it's even available for transcription to begin. And then the process of transcription can be controlled by transcription factors. We can have enhancers, we can have repressors. The actual processing of the mRNA, does it get spliced correctly? Is the tail and the cap added? Does it leave the nucleus? Then the translation process. How long does that mRNA stay before it's degraded? Is it recognized and able to be translated by the ribosome? And finally, post-translational modifications would mean once that ribosome completes the production of that protein, it may not be functional as is. It may need to have a little part of it cleaved, or it may need to combine with another protein to be functional. So those things would regulate whether or not we have a functioning protein product. Now let's look at the Lac Operon. So there's a few things that I want us to understand before we get into the figure. First of all, lactose is a sugar, and a bacterial cell, just like us, can use it for energy source to produce ATP. So in other words, we can metabolize it, use it as an energy source. Okay, the Lac Operon codes for genes that instruct for enzymes. So an enzyme, remember, is the protein. That's the protein product that would metabolize lactose sugar. So in other words, the Lac Operon is controlling whether or not these enzymes get made that can actually break down lactose. Well, transcription is blocked by a repressor when lactose is not present. And that would make sense, right? Because why would I want to spend cellular energy making enzymes that break something down that I don't have? Now, when lactose is present, it can actually bind and change the repressor so that the repressor no longer can prevent transcription. Now we have one more thing we will talk about. We're going to go through that. I'm going to draw a line right here. We're going to go through that part of it, and then we'll talk about this bottom part and why it's important. So here's our Lac Operon, and what we see is right now, so here's the promoter. Okay, that's where the RNA polymerase is going to bind, right, and would normally start transcription. However, in this Lac Operon, we have an operator region. The operator is where a repressor can bind. And what we see is that's exactly what's happened. We have a repressor that's sitting down on top of the operator. Well, RNA polymerase can't pass that. Therefore, these particular enzymes that break down lactose will not be produced. Now, when lactose sugar is in the environment where the bacterial cell is growing, it can actually bind to the repressor. It changes the shape. It no longer will remain bound to the operator. Now, RNA polymerase can go ahead and transcribe those genes. As long as they get translated, then the cell is going to be producing the enzymes that will actually metabolize or break down lactose. Now, that is all true, but it turns out that that really only happens in very small quantities as long as glucose is present. So in other words, glucose would be the preferred sugar, right? As long as there's plenty of glucose, then this is not going to happen. But there is this protein called CAP, and it is activated by something called cyclic AMP. And what I want you to think about with these is quite simply, these are the hungry signals. So when glucose gets low, these are going to be around. So what this does is this actually increases the amount of transcription that's occurring. So in order to get any measurable amount of these enzymes, then two things have to be true. We have to have lactose present. That removes the repressor. And we have to have glucose absent so that these hungry signals will bind and they will act as transcription factors that will promote more transcription. And that is the Lac Operon, and that is one way that prokaryotic cells genes are regulated. Now we talked about eukaryotic gene expression, how complicated it is. And remember, in each one of these steps, all of these steps can be regulated so that if there is a stop at any of these steps, then we don't get to the ultimate protein product.